Hello and welcome, everyone in the ICH Guideline video series. This video will provide you a detailed outline on ICH Q3C Guideline on the residual solvent impurity. Let's get started. The ICH Q3C Guideline addresses the permissible levels of residual solvents in pharmaceuticals. Residual solvents are organic volatile chemicals that are used or produced in the manufacture of drug substances, excipients, or in the preparation of drug products, but are not intended to remain in the final product. They are typically left over from synthesis or other processes and can potentially remain in the final pharmaceutical product in trace amounts, depending on the purification methods employed. Established by the International Council for Harmonisation, ICH, it categorizes solvents based on their toxicity and provides recommended limits for each. The guideline aims to protect patient health by minimizing solvent-related impurities in drug products. The guidelines provide a harmonized procedure to set acceptable limits for residual solvents in drug substances and drug products based on their permitted daily exposure (PDE) limits. Permitted daily exposure (PDE) denotes the safe amount of a potentially harmful substance an individual can be exposed to daily over their lifetime without adverse effects. PDE is used in the pharmaceutical and regulatory industry to assess the acceptable level of exposure to a particular drug substance, such as a residual drug or a potentially harmful impurity. PDE values are expressed in milligrams per day and concentration in parts per million, ppm. The method used to establish permitted daily exposures for residual solvents is presented in Appendix 3 of ICH Q3C Guideline. The calculation of PDE involves 6 steps, and the steps include: determine the substance for which you need to calculate the PDE, collect data on the substance's toxicity, pharmacokinetics, and any relevant safety studies. The reference dose is often derived from toxicological studies. It is the highest dose at which no adverse effects, NOAEL, no observed adverse effect level, or the lowest dose at which adverse effects are observed, LOAEL, lowest observed adverse effect level, occur. To account for uncertainties and differences between animal and human responses, various uncertainty factors are applied. These factors could include interspecies variability, intraspecies variability, and others. During PDE value calculation, these uncertainty factors are considered, where F1 is a factor for extrapolation between species, F2 is for variability among individuals, F3 is for study duration, F4 is for severity of toxicity, and F5 is used if the no effect level was not established. Depending on the regulatory guidelines and the nature of the substance, additional factors may need to be considered. For instance, if the substance is a genotoxic impurity, additional calculations might be necessary to determine an acceptable limit. After calculating the PDE, it should be compared with the estimated or actual exposure levels. If the estimated exposure is below the calculated PDE, it suggests that the level of exposure is considered acceptable. ICH Q3C Guidelines provides 2 options for calculating the limits of residual solvents. Option 1. Identify and quantify all residual solvents present in the drug substance or product. Option 2. Identify and quantify solvents known to be used in the synthesis or purification of the drug substance. ICH Q3C has categorized residual solvents into 3 classes based on their risk assessment. Class 1 solvents to be avoided. These solvents are known to be carcinogenic, mutagenic, or have other severe toxicities. Given the risks they pose, it's recommended that they not be used in the manufacture of drug substances, excipients, and drug products unless their use can be justified and their presence can be limited to acceptable levels. The examples of Class 1 solvents are benzene, carbon tetrachloride, dichloroethane, trichloroethane, etc. Class 2 solvents are not as harmful as Class 1 solvents, but they still have inherent toxicities that should be limited in pharmaceuticals. They should be used in pharmaceutical manufacturing only when necessary and when no safer alternative is available. Their concentrations should be limited to established permitted daily exposure levels. The examples of Class 2 solvents are acetonitrile, chloroform, methanol methylene chloride, tetrahydrofuran etc. Class 3 solvents have low toxic potential to humans when used in pharmaceuticals. They may be used in pharmaceutical products, but their amounts should be controlled and minimized for good manufacturing practices. For examples, butyl alcohol, ethanol, propanol, isopropyl alcohol, etc. ICH Q3C guidance document doesn't dictate a specific method for the analysis of residual solvent, but it should be validated. Gas chromatography is mentioned as an effective method for detection and quantification of many residual solvents. Details on solvents used, reasons for their use, methods for removing solvents, and testing methods employed to determine levels should be provided in the marketing authorization dossier. In wrapping up our exploration of the ICH Q3C guideline, it's essential to recognize the paramount importance of understanding and adhering to these standards. Ensuring the safety and efficacy of pharmaceutical products hinges on our commitment to limiting residual solvents. By diligently applying the principles and best practices outlined in this guideline, we not only uphold the highest scientific standards but also ensure the well-being of patients worldwide. Remember, integrity in our processes leads to trust in our products. Thank you for joining this training, and let's continue to prioritize safety in all our endeavors. To get details specific to ICH Q3C, you would need to refer to the official ICH documentation or other relevant sources which have the latest versions of these guidelines.